Okay, welcome everybody. I am Carola-Bibiane Schönlieb. I'm one of the co-directors of the Center for Mathematical Imaging in Healthcare, and I hope we have a very interesting day in front of us, I think. I'm delighted that so many of you could make it, and before we really start with the core of this meeting, the actual academic and industrial presentations, I thought I'd just give you a brief primer on what the center is all about, who we are, what we do, and what in particular the research is that we are carrying out in the center. So first of all, we are one of 5 EPSRC centers for mathematics in healthcare that have been developed over the last 2 years or so. We are focusing our work in a collaborative fashion between mathematicians, statisticians, engineers, physicists, biomedical researchers and clinicians to develop analysis, processing, reconstruction techniques for imaging data that arises in different contexts in the clinic, in neurology, in cardiovascular imaging, and in cancer imaging in oncology. And the main philosophy we are working on is really to find projects and problems that can benefit from this multidisciplinary approach of mathematicians, statisticians working together with people in the clinic. So here are the investigators in the center, and again they reflect the range of disciplines that are involved, which range from here mathematics to the clinic. Maybe I'll just point out a few people here. So John Aston, who is the co-director, who can't be here today. He's actually seconded to the Home Office at the moment, so but still involved in the center. James Rudd, who is our clinical lead, who is sitting there in the back. And then there are various other people. Raj is not here yet, but he will give a talk later on. And then there are also other people who are not on the slide who are somehow affiliated with the center. So for instance, Ursula, whom I have seen before, who is here, who works on elastography, and then apart from the academic investigators, we are also lucky to be supported by a large network of industrial partners with whom we have ongoing and underway collaborations. And again, some of them are represented here today, like Toshiba and GSK, who are also chairing sessions later on. The core of the center are really the postdocs and the PhD students. And here you see them represented. Again, what they're doing will become clearer in the course of today, because some of them will give a presentation later on. Some of them have posters in the poster session, so please have a look at what they're doing. What I want to do now is just to give you a few examples of projects the postdocs are working on. Okay. So I start with Angelica, who joined us in August, I believe, this year. Yeah, July. And Angelica works on spatiotemporal image reconstruction, in particular on the aspect of having fast imaging techniques, hence high temporal resolution, while keeping also the spatial accuracy to a certain extent. And so the idea here is, for instance, in magnetic resonance tomography, what enables you to image faster in time is that you acquire less data for each time step, which in this case, in MRI is in K-space, are Fourier measurements. And so if you acquire less data for each time step, of course, you would think that you have lower spatial resolution. But you can play around with this a little bit. And here the idea of what Angelica is working on is to complement the measurements that you take that are now undersampled by some additional information, which in her case is motion. So what you see here is a graphic which shows the effect of informing the reconstruction of a 4D imaging data set with a motion model, where this graphic here is quite complex, but let me briefly talk you through this. Angelica will also present this later on, so she can give much more detail on that. But here the idea is the following. So here on the left, what you see is a kind of typical compressed sensing pipeline, which uses ideas of sparse reconstructions in the spatial domain to still aim to reconstruct high-resolution images, even if you have not sampled everything in K-space. And so the phi here that you see corresponds to the amount of undersampling that has taken place. So phi equals 0 means we have all the Fourier measurements and we reconstruct from them. So this is a one 2D slice out of this 4D imaging data set. And once phi increases, phi tells you how much undersampling you have done. So the larger phi, the less measurements you have taken for each time step. And so this is the kind of one of the by now standard pipelines, this compressed sensing reconstruction approach, showing you the results for different levels of undersampling. And here on the right you see the same mechanism, but now a compressed sensing motion-informed reconstruction, where you also not only use sparsity spatially, but you also use a dynamic model which correlates the information in the fourth dimension in time. And so visually you do not see a lot of difference in the pictures, but if you look, think of this as some kind of error plot. So this is a particular measure for image quality, which is called the structural similarity index, which compares the undersampled reconstructions with the fully sampled reconstruction. And what you see here, and I should say red means that's good, so the quality is very good. Blue means it's not. And so what you see here are the plots for increasing amounts of undersampling of both the compressed sensing type reconstruction and the compressed sensing motion-informed reconstruction. And what you see is that the quality is much better preserved if you also include the motion as an additional information in this reconstruction process. Okay. And here you see the same thing, just boiled down to one scalar error measurement over various slices. So you see this is a consistent phenomenon where the green line is the quality of the compressed sensing motion-informed reconstruction. Okay, Milana is another postdoc in the center. Milana works on two different aspects. One, again, is related to what we've seen before on the reconstruction process of imaging data. In particular, she looks at endoscopic imaging and is concerned with the problem of when you do endoscopic imaging of, let's say, the gastrointestinal tract, then you're putting a cable down the body, and inside this cable there are fibers which are entangled from the point where actually the signal comes out to the point where it's measured. And so this entanglement process has to be modeled mathematically in the reconstruction process. And if you do this well, then you can go from a raw measurement where the entanglement hasn't been solved to an image where you can actually see something. Another thing that Milana is working on is high-dimensional imaging data. And here the idea is, think about multispectral imaging, for instance. The idea is that usually you're not interested in the multispectral images directly, but in extracting some low-dimensional features from them. And what she's doing here is to develop methods that can do this in a statistically robust way for detecting and extracting important features in high-dimensional data sets. Parham also just joined us. He's working with Thanasis Fokas in the Applied Math Department, and they are looking at reconstruction of current in EEG with methods that are direct, in the sense that the only thing that is being done to go from the measurements to the current is a matrix inversion, very roughly speaking. So this is a very fast process. It's also called analytic inversion technique. And what they show here is that for most of the sensors, and actually this plot has been very much blurred, because you can't really, so here you can't really see the difference between the predicted and the measured currents, so they're very close. And for most sensors, this is true. So this is a very accurate and fast inversion technique for EEG. Yuan will also speak later today. Yuan works with James on extracting mechanical models from imaging data. So here the imaging data is a mean to deploy mechanical models in cardiovascular in a cardiovascular system to find out what is the correlation between certain mechanical characteristics and certain types of cardiovascular diseases. So, for instance, in this first project, what they're interested in is to find out what is the correlation of mechanical stress extracted from CT images, as well as FTG uptake extracted from PET images, to the expansion of aneurysms, where in this particular project they have been looking at a patient cohort which has been seen once and then 2 years later, and then this has been studied over this time period. And Yuan will talk more about this. Sorry, now I need some water. We'll talk more about this later. Okay. Okay. So I haven't mentioned machine learning yet, and I think it's about time. Every one of you is waiting for me to say that. So what we are doing here in the center is to look at mathematical foundations of machine learning, in particular applied to medical imaging. And here Pan is also a postdoc who joined us even more recently, like a month ago or so. And Pan looks at particular mathematical models which are called variational regularization techniques that can be used for various imaging tasks like image reconstruction, denoising, motion estimation, segmentation, whose solution very much depends on a good parameterization, on a good setup of these models. And to find such good setups, to find optimal parameterizations of such approaches to perform these imaging tasks, One particular type of machine learning approach that people were looking at over the last couple of years is so-called bi-level optimization. And certain questions that Pan is concerned with is the robustness of these optimal parameters. How optimal are they? Can we mathematically prove that they're maybe even globally optimal? Can we employ fast numerical solvers to train these variational approaches? And these are the questions. Sorry, there's a lot of mathematics here on the slide. These are the kind of questions that that Pan is concerned about. To go, for instance, to solve imaging tasks like this, where you have a noisy image and you want to get rid of the noise in an optimal fashion. U works again on the foundations of machine learning, developing deep learning and statistical techniques for supervised and unsupervised learning. And one of the things that is also visualized here on the right, sorry, I have a cold, and that's now showing. That is visualized here on the right is the detection of abnormalities in an unsupervised fashion without human annotation. And so this, I believe, is an example of this Brett's 2015 brain data set, where you can see the original, and in this case we have a ground truth, and this is a result that you obtained by an unsupervised approach of detecting the abnormality in this brain scan. Okay. Jingjing Zhu just joined us also. There are lots of newcomers, so that's why this is really an update on center activities. Jingjing works on two aspects in medical imaging. One of them is the analysis of longitudinal imaging data, where she looks at high-dimensional imaging data such as fMRI and time series data of that, and wants to extract a low-dimensional representation of active regions in the brain, for instance. Another thing that Jingjing is working on is the analysis of imaging data on manifolds, and again of high-dimensional imaging data or functional imaging data. And one of the things that one particular approach she's looking at is tensor regression on manifolds, for instance, to deploy unsupervised and supervised studies of the geometry and functionalities of the cerebral cortex of the brain. Okay, and then we also have, I said, we have the postdocs and also the PhD students, and here again just a few examples of what the students are working on. Joanna actually has just submitted her thesis recently, but what she has been working on was the development of image analysis techniques for cellular imaging, in particular for cell tracking from microscopy images. Erland works on super resolution for photoacoustic tomography. And Eardi works again on functional imaging, and again functional imaging on the cerebral cortex, in particular focusing on the fact that informing the functional data analysis with the geometry of the cortex gives you much better results, much more accurate results than not taking it into account. And last but not least, Veronica works on what is also called microstructure MRI imaging, where she wants to use structures extracted from microscopy imaging, tissue structures, to enhance and inform the MRI reconstruction, such that the MRI reconstruction also shows some of the features that are shown that turn up in the microscopy images. Okay, so 2 more slides, one on our outward-facing side. So we have a partnership fund in the center, which we very much want to use to get in contact and to broaden out the things that we are doing, to also help other researchers who are not in the center to realize some of their ideas, research ideas that they have. So there is seed funding in this partnership fund. Funds you can apply for to organize workshops, funds to bring intern students here, for instance, or for students to go somewhere else. And if you want to know more about this, please look at our website, where you get the details of how to apply for these funding opportunities. And a very new thing that we have started last week, actually, is the imaging clinic. So this is not a clinic where you go if you have some health problem. I mean, you personally have a health problem, but this is a clinic for data problems, for imaging data problems, both you know, on the image acquisition side to the analysis and processing side. And so these imaging clinics, they happen fortnightly on Wednesdays, alternatingly between the cafeteria in CR UK, down at the Edinburgh site, and here in the Center for Mathematical Sciences. And the idea is really, if you have, you know, either you have a problem and you don't know exactly where to go or what to do, you have a certain aspect you want some advice on, you can come there with your data, with your question, or maybe you have, you know, a more open-ended suggestion for maybe a collaboration with someone in the center. So all of these, you know, ranges between very detailed questions to more visionary ideas are welcome in this clinic. Okay, and I think that's it. So, without further ado, and I think I'm still on time. That's good. I thank you very much for your attention, and I hand over to Ian. Yeah. For the first session.